Hello students, I am Akshay Deshpande and welcome to biology class. And in this class today we will be beginning a completely new chapter that is biomolecules. And biomolecules is fairly an important chapter. I can say this chapter is a lot of biochemistry. Okay, this is mainly a kind of biochemistry chapter where the intention of the whole chapter is mainly to introduce about the molecules that are involved in the living system and what are their purpose and what do they do in the living systems. That is what the main purpose of the biomolecules is. And this is the first class, as you people all can see over here. And today we will be starting with the first part of the chapter, that is how to analyze any chemical composition in the living systems. Right? So let's get started then. Now, if you look into our planet and if you take any living organism, okay, what we mean by over here is any tissue, okay, any tissue if you try to take from any living organism, and also if you try to analyze the contents of the earth crust, okay, mainly with respect to we can say the soil samples, right? If we take soil samples, right, and if we analyze it with respect to by comparing the contents of the living tissue and also by comparing the contents of the soil samples, you will see that there is no much difference with respect to the constituents of the tissue and also the constituents of the earth crust, right? For example, the major of the contents that you will see over here are the carbon, the oxygen, the next one we are having is the hydrogen, and finally some other similar lists, right? So we can say that we are no different than any other constituent of the planet Earth. Ultimately, we are made up of all the basic compounds or the minerals, we can say, that is carbon, oxygen, hydrogen, and some other inorganic constituents. So this is mainly about the comparison of the living systems and also the earth crust. And here we are actually having a table which shows the different elements, okay, different type of elements, and also the percentage of weight of the earth's crust of these constituents and also in the human body. Right now I have mentioned the table directly from the textbook, and I have seen that some of the questions do have appeared from this particular table, so we can say that this table is quite important for our discussion, right? Yes. The first element we can see over here is the hydrogen, and the percentage in the case of earth crust is around 0.14 percentage, and in the case of a human body it is only 0.5 percentage. On the contrary, you can see that the carbon is around only 0.03 percentage in the earth crust, but whereas in the case of living tissues of human body, you can see it is around 18.5 percentage. And also in the case of oxygen, you can see that it is one of the most abundant element present in the earth crust, that is around 46.6, and in the case of human body it is around 65 percentage, right? So if you look into all other constituents, you can easily notice that the oxygen is very important in the terms of the percentage of weight in the earth crust and also in the human body, right? So in the earth crust the most abundant is the oxygen, and even in the case of human body it is the abundant. But when you compare between the human body and also the earth crust, you can clearly see that oxygen is more in the human body than even in the case of the earth crust, right? The nitrogen is very little over here, right, in the case of earth crust, but whereas in the human body it is around 3.3 percentage, and sulfur is around 0.03, same as the carbon, and 0.3 in the case of human body, and sodium is around 2.8 percentage in earth crust and 0.2 in human body, and calcium we are having 3.6 and 1.5 in the case of human body, and magnesium is 2.1 and 0.1 in the case of the human body. Silicon we are having around 27.7 percentage in earth crust, but however the same element in the human body is completely negligible. right? So now they have mentioned all of these elements, okay? And if we have to remember the important ones, okay, if you have to emphasize the most important ones from all these mentioned elements, we can say that try to remember the major constituents, major elements, okay, major element percentage, right? See, if you consider the earth crust, you can clearly see that oxygen is the major. The next comes we are having is the silicon, right? And the next one we are having is the calcium, right? So try to remember the first top three major constituents in the earth crust, okay? And also try to remember the least values, okay? So if you consider the least value, we can say that the nitrogen is completely very little, okay? You can try to remember this. And carbon, and finally we are having is sulfur. These three are the least, okay? So highest we are having is the oxygen, and silicon is highest, and finally we are having that is the calcium being the next highest, right? The top three. But the nitrogen is the least, okay? And also the carbon is the least in the earth crust, and also we can say that the sulfur is also least, right? The up arrow mark indicates the highest, and the down arrow mark mainly indicates the lowest one. And similarly, if you compare with the human body, try to remember the highest values. As you people can clearly see, that carbon, oxygen, and the next one we are having is the nitrogen, the three clear winners for our remembering, right? The first one we are having is the carbon. The highest quantity we are having the oxygen, the second we are having is carbon, and the next highest we are having is the nitrogen, okay? And also now in the case of human body, if you try to recall or try to remember the least constituents, you can see that silicon is the least constituent. Next comes we are having is magnesium, and the next one we are having is the sodium, right? So try to remember these three highest and these three lowest, okay? So that is how the questions are generally framed, that they will mainly concentrate on the major elements and also the minor constituents, okay? So you don't have to worry about the intermediate values. Nobody is going to ask you about the intermediate values, okay? And in this way you can just try to make a small table for your reference and try to recall them every day as much as possible, okay? Ultimately this we have to try to make a chart, make a concept map, and recall them as much as possible, okay? That is how you can make a firm confirmed memory regarding this, okay? We have seen, let me tell you, like I have seen many questions where they have asked which of the following is a minor constituent or a major constituent or completely negligible or very little. They have asked this, okay? So such kind of questions are direct questions which will yield you 4 marks, right? So try to, as I said, try to make a note on these list of elements that I have mentioned in your textbook. Now let us go on to the next one. That is organic compounds in the case of organisms, right? So now how can we determine the organic constituents in the tissues of the living organisms? It is quite a simple task if you ask, because we can take a tissue. If you want, you can take plant. It is very easily accessible and even in some cases you can even take the animal tissues also, right? And we have to grind this particular tissue that we have taken with a chemical called as the trichloroacetic acid, okay? We have to grind it with the trichloroacetic acid, right? So they have mentioned the formula Cl3CCOOH. This is the chemical formula for the trichloroacetic acid, right? So you have to grind it in the mortar and pestle, okay? Mortar and pestle, right? If you grind it in this equipment, you will obtain a thick paste like a semi-solid slurry, okay? And we have to strain it, okay? We have to strain it. That is straining is a general process that you people have seen in your household, right? We have to take a cloth and place that cloth in a bowl or a container and put that particular slurry in this cloth, that is a muslin cloth, and what happens is that you will land up with the liquid substance, and also you will land up with the solid substance, right? So now this filtrate we are very much concentrating, right? We are getting mainly two types of fractions. The first one we call it as the acid soluble fraction, which is mainly present in the filtrate. Okay, this acid soluble is mainly present in the filtrate, but whereas the acid insoluble is mainly present in this the cloth leaving what we get. So the acid insoluble and acid soluble, why they are called as in that way? Because the type of organic solvent we are using over here, that is the trichloroacetic acid, right? So all the organic molecules which are present, they will be present in the filtrate, and the filtrate is mainly made up of the trichloroacetic acid, right? So now we can actually determine the constituents in the acid soluble fraction. Okay, and we are having a technique called as the chromatography, right? So chromatography, I hope you people can remember from your first year practicals. There's a demonstration called as the paper chromatography, right? We are mainly using the Whatman filter paper, and we use an organic solvent in a cylinder or a big jar, and we will dip our chromatographic paper in the jar in such a way that only a tiny tip of that particular paper touches the buffer or the organic solvent, right? So due to the capillary effect, what happens, the solvent will actually rise through the paper, and the paper that is containing the mixture of solvents or the substances will gradually separate based upon the affinity. Those substances will show towards the organic solvent that we are using, right? So hence we can say the chromatography is used for the purification and separation, right? There are many other techniques. For example, we are having centrifugation, right? It is mainly used for separation but not for purification, right? And also we are having electrophoresis. It is also used for separation but not for purification. Remember, both with separation and purification, we are having a technique called as the chromatography, which is widely employed in various laboratories. So now, using these technique, that is mainly the chromatography, we obtain the molecules which are present in the living systems called as the biomolecules. And in a simple way, in the most briefest way to define a biomolecule, we can just simply call them as a carbon-derived molecules. We can say that it is mainly a carbon-derived molecules, right? So now these are the carbon-derived molecules. But what about the inorganic nutrients? Inorganic nutrients. Can we identify the inorganic nutrients? Yes, we can identify the inorganic nutrients, but we have to do a destructive experiment for this. Destructive experiment. Right? So what do we mean by a destructive experiment? It means that we have to take the tissue, dry the tissue, and after drying the tissue, we have to burn it. Right? So the intention of drying is mainly to remove the water content in the tissue. Right? Hence we are drying it and we have to burn it, and finally the remains of the tissue is called as the ash. Right? And if you analyze the ash contents, you mainly find inorganic nutrients. For example, calcium. For example, we are having magnesium, okay, and sodium, right? So on. All of these we can say that inorganic nutrients, and interestingly, even these inorganic nutrients are also coming under the fraction called as the acid soluble fraction. Yes, acid soluble fraction. So now we are having ultimately organic constituents, we are having inorganic constituents, and also we are having the functional groups, right? Now, for example, the functional groups, we are mainly using it for the classification. Okay, we mainly try to determine the functional groups, okay, from the chemistry point of view. From the chemistry point of view, right? So here we are mainly concerned with the functional groups, right? But in the case of biological point of view, from the biology point of view, we can say that we just classify the biomolecules, and that is whether they are amino acids, right? Whether they are amino acids or they may be carbohydrates, right? They may be carbohydrates, right? Or they may be proteins, and etc. Right? So simple classification, we mainly discuss it from the biological point of view, and if you are looking into the functional groups, generally we discuss it from the chemistry or chemical property of the substances or the molecules. Right, so we have made the concept quite clear over here. So now let us look into the first molecule, that is the amino acids. Now, amino acids are the building block of the proteins. Okay, yes. So if you look into any type of protein, ultimately they are made up of the amino acids, right? Yes. Now, why they are called as amino acids? Because they do have a very interesting property, that is the amino group, which is actually basic in nature, but whereas the same molecules also has another group, that is the acidic group. Okay, acidic group, and this acidic group is generally the carboxyl group in the case of the amino acids, right? So also you find simple R groups. You also find R groups, right? So we will discuss about the amino acid in the structural point of view. Okay, so if you have to draw a simple structure of the amino acid, it is NH2 C, that is carbon, and H, and here we are having the R group, and finally we are having is the carboxyl group, right? So this is the, what we can say, general formula, right? This is the general formula for the amino acids. Now here you can see that this carbon, what we are mentioning over here, is called as the alpha carbon. Okay, the reason why it is actually called as the alpha carbon, because you can see there are 4 different groups attached to this single carbon, right? So hence, as mentioned over here, it is an as amino group, which is basic in nature, and also the acidic group, and it has an alpha carbon, and for this alpha carbon, the 4 different valence groups are actually attached, right? For example, the first group you can see over here is the simple hydrogen, and the next one we are having is an amine group, that is the NH2 group is called as the amine group, and this group is mainly giving the basic property to the amino acids. And also the last one we are having is the carboxyl group, that is COOH is actually the carboxyl group, and this carboxyl group is giving the acidic property to the amino acid, as you can look over here, right? And here we are having one another group called as the R group, right? See, why do we use the alphabet R? R can be any group. Okay, it is just an alphabet that we're using to indicate presence of any group over here, presence of any group over here. Remember this, right? See, in some cases you find a simple hydrogen. Okay, let me draw or write one amino acid over here. Okay, NH2 alpha carbon, hydrogen, and carboxyl group, right? And let us substitute this R value as a hydrogen, right? So if you find a simple hydrogen, a simple single molecule of hydrogen in the R group, then this amino acid is the glycine, right? Now glycine is considered to be one of the simplest amino acids among all other amino acids, right? The first simplest amino acid that you can learn to study, right? We can even glycine, in the case of glycine, it is having simple hydrogen in the case of R group, right? So now in the case of another amino acid, if you consider alanine, okay, in the case of alanine, the R group is simply methyl group, right? That is CH3 group is actually a methyl group, right? And in the case of serine, okay, in the case of serine, you can see it is the R group is CH2OH, right? CH2OH, which is an hydroxy methyl group. So with the same skeleton of the basic formula of amino acid, you can just go on replacing the R group with the desired substituent group. You will get a different type of amino acids, right? So generally you see that in nature the amino acids are around 20 in number, Okay, so whatever proteins you consider, okay, so ultimately they are made up of the repetitive units of these 20 amino acids only, right? And the R group basically varies. So let us look into some other chemical properties of the amino acids, right? See, for example, we are having some amino acids which are charged, okay, which are charged. For example, there may be a positively charged or they may be what? Negatively charged amino acids. An example for negatively charged amino acid is glutamic acid. It is glutamic acid, right? And negatively charged amino acids are generally called as the acidic amino acid. But the positively charged amino acid, we are having an example called as the lysine, right? Lysine is an example for positively charged amino acid, and positively charged amino acids are actually called as the basic amino acids. And in some cases, you also find neutral amino acids. You also find neutral amino acids, right? An example for a neutral amino acid we are having is the valine. Okay, valine is an example for the neutral amino acid, and in some cases you also find aromatic, okay, aromatic amino acids. We are having 3 examples that has been mentioned in your textbook. The first is the tyrosine, okay, tyrosine tryptophan, okay, and we are having one more, that is the phenylalanine, right? It is a very simple amino acid, alanine with a phenolic group. It gives us the phenylalanine. The name itself indicates, just attach a phenolic group to the R group, then you get phenylalanine. So this is some basic examples that we are having regarding the amino acid types based on the chemical properties of the amino acids, right? Now there is also an interesting property, that is the amino groups and the carboxyl groups are actually ionizable, okay? What do we mean by ionizable? In the cells, they can give up the protons from their amino groups and the, what we can say, the carboxyl group, and attain a particular charge, okay? Either they may be net positively charged, or either they may be net negatively charged, or they may be having no charge at all, okay? They may be having no charge at all, right? Now, out of all these types, that is, it may be carrying a net positive charge or a net negative charge, the most interesting point that we have to consider is about the molecule that is having no charge at all, right? And the amino acid which is carrying no charge at all is called as the Zwitter ion, right? So let me erase this and let me draw the what we can say, the 3 different charged molecules or the ways of the amino acid. For example, we are having this one, and alpha carbon and R group, and we are having hydrogen and carboxyl group, this one, yeah. So next we are having one more structure, COO minus, yeah. So we are having R group, and here we are having the H, and finally the last structure that we will be writing is this one, COO minus, H group, and then R group, exactly, right? So this is what they have mentioned in your textbook if you look over here. So now this is an A molecule, consider, and this is a B molecule, and this is as the C molecule. And now if we try to look into the net charge that this particular molecule possesses, be it A or be it B, be it C, whatever, okay? So you may expect a question where they will mention about the amino acid. They may draw and give an amino acid, and they may ask you what is the net charge of this particular amino acid, okay? See, the calculation of net charge of the amino acid is way more complex. It's completely, what we can say, out of the scope of the current discussion. But based on the diagrams or the molecules that they have mentioned in your textbook, we can just draw, or we can just come to a conclusion about the charges over here, right? See, now the A molecule is having only one positive charge, but no charge in the carboxyl group. Hence, we can say it is a plus 1 is the charge of this particular A molecule. But now if you look into the B molecule, the net charge of this particular form of the amino acid is going to be 0, because you see that it is having both the plus charge and one more we are having is the minus charge. Both the charge will get cancelled, hence we say it is the net charge of this B form of amino acid is going to be 0, right? And the last C diagram what we have drawn over here, the structure of the amino acid, there is no positive charge on the amino group, but however the carboxyl group is carrying one negative charge, hence the charge is going to be -1, right? So in this way, from the diagram or the structures that they have mentioned in your textbook, you can expect one question, right? Identify which of the following is a zwitterion in this way, or they may even ask you, or they may give the structure of that particular amino acid and they may ask you the net charge, right? So don't worry, they're not going to ask you beyond the scope of whatever they have mentioned in your textbook. Ultimately the questions are going to come from the mentioning part of your textbook itself. Okay, so don't have to bang your head regarding trying to learn advanced concepts. Okay, you will indeed study the advanced concepts as you take on the higher classes. Yes, so this is mainly about the amino acid, right? So out of these three structures, that is A, B and C, the B structure is called as the zwitterion, right? That is the net charge of this particular molecule is 0. I hope I have made quite clear regarding the zwitterion, and it is an important point that you people have to remember among the amino acids, right? Examples for the amino acids based upon the charge group or something like that, remember those, okay? And also try to remember about the zwitterion. Yeah, so just to try to draw these structures much more and more frequently, you can see that you will try to remember this in a better way. The next one we can look over here are the lipids. Okay, now lipids are actually water insoluble, right? See, we have studied about the lipids, and one of the important physical chemical property of the lipids is that they are completely insoluble in the water, right? Yes. So now some lipids can be simple fatty acids. Okay, some examples for the lipids may be simple fatty acids. Okay, and generally to the R group, you see that the carboxyl group is actually attached. Okay, the carboxyl group, that is COOH group, is attached to the R group, and based upon the number of carbons, okay, based upon the number of carbons including the carboxyl group, an example they have been mentioned over here, that is the palmitic acid. It mainly has around 16 carbons, right? If you have tried the structure of palmitic acid, it is CH3, CH2 and 14 COOH, right? So this is, sorry, yeah, so this is the formula for the chemical formula for the palmitic acid, right? And also we are having one more that is called as the arachidonic acid, and it is mainly made up of 20 carbons. So in the case of both palmitic acid and also the arachidonic acid, remember we are considering both the carboxyl groups over here. Okay, considering both the carboxyl group, it is mainly having 16 and 20 carbons, right? So they may ask you how many number of carbons are present in the arachidonic acid or palmitic acid. Remember, always the number of carbons are going to be even in number, never the odd case, right? Yes, this is one simple way to remember it. Now arachidonic acid, if you try to recall the formula, it is a CH3, similar to that of the palmitic acid, CH2, and we are having 18 and COOH, right? So 18 carbon from the CH2 and 1 from the CH3 and 1 from the COOH will completely make us 20 carbon. Same holds true for even palmitic acid. It makes the 16 in the case of palmitic acid, right? Yes. So now in some cases they may be saturated or they may be unsaturated. For example, palmitic acid is an example for a saturated, and arachidonic acid is an example for the unsaturated, we can say, fatty acids. Right now, another simple molecule or a simple lipid we are having is the glycerol, right? We are having glycerol as another example that they have mentioned over here. And if you try to look into the structure of the glycerol, it is a CH2 OH, CHOH and CH2 OH, right? Hence it is called as the trihydroxypropane because of 3 hydroxyl molecules present in the chemical structure, right? So it is called as the glycerol. The alternative name we are having is the trihydroxypropane, right? For a propane molecule we are having 3 hydroxyl groups, hence it is trihydroxypropane in a simple way, right? And glycerol are very much important. Okay, so now in the case of glycerol, if you esterify the glycerol, okay, along with the fatty acids, okay, we get some interesting molecules, okay, that we can look over here. Fatty acids are actually esterified with the glycerols, okay, and the result of esterification of fatty acids with the glycerol may yield us mono, not a two, it is a di, okay, mono, di and a triglycerides, okay, mono glycerides, diglycerides or the triglycerides, right? And another type of lipid we are having is the oils, okay, and generally oils are having a lower melting point than the fats, hence in even colder temperatures they remain as oils, right? An example for the oil is the gingerly, okay, sesame oil, we call it as a sesame oil, called as the gingerly, right? Yes. Now we are having one more type of lipid called as the phospholipids, right? Now if you try to recall the term phospholipids, we have mainly encountered this term in the where cell membrane, right? If I'm not wrong, in the cell membrane you have studied about the phospholipids, right? And one example for the phospholipid we are having is the lecithin, or the another name for lecithin we are having is the phosphatidylcholine, okay, phosphatidylcholine, okay, an alternative name for the lecithin, remember this, right? Not required though, but still as an extra point to remember, okay? And why they're called as phospholipids? Because along with the fatty acids, right, plus glycerol, right? So what is the structure we get? Fatty acids plus glycerol, we get actually the glycerides. Okay, remember if there are 3 fatty acids, we call it as a triglycerides. If it is a single fatty acid, it's called as monoglyceride. If it is made up of 2 fatty acid chain, then it is called as the diglycerides, right? So along with the glycerides, that is the fatty acid plus glycerol, we get the glycerides, right? So if I attach a phosphate molecule, okay, if I attach a phosphate molecule, the ultimate result that you're going to get is the phospholipids. Yes, phospholipids, right? Yes. And one such example for phospholipid we are having is the lecithin, as we just mentioned a moment ago, right? So the phospholipid, that is lecithin, is having one more group called as the choline group, okay? Hence the name is phosphatidylcholine. Okay, so let's not look into the more detailed structure of the phosphatidylcholine or lecithin, right? So now in some cases, that is in the neural tissues, in the nervous system, you do find more complex lipids, okay? The more complex structural lipids are actually found in the, we can say, neural tissues. For example, ceramide, okay? We are having ceramide, or we are having sphingolipids, right? Yes. Ceramide is an example for the sphingolipids, okay? Ganglioside, right? Cerebroganglioside, so on. There are many examples, but generally sphingolipids are mainly found in the neural tissues, and sphingolipid is an example for the complex lipids. Example for complex lipids. See, I hope you people might have heard of the one more term that is called as the glycolipids, okay? Glycolipids, okay? Try to recall which cell organelle is involved in the production of glycolipids and glycoproteins, right? I will not mention it over here, but you people have to try to recall regarding the site of synthesis of the glycolipids, okay? Glycolipid is also found in the nervous system, okay? And it is also an example for complex lipid, right? So some complex lipid structures are also found in the nervous tissues. So now we can look into the last part, that is the heterocyclic compounds. Let me erase this once again. Yeah, we are having is the heterocyclic compounds. Now, heterocyclic compounds are mainly we call them as the nitrogenous bases. Okay, so if you remember the nitrogenous bases, we are having the purines and the pyrimidines, right? We have studied about the purines and pyrimidines in our molecular basis of inheritance, that is the DNA, about the structure of DNA, right? So in the case of purines, we are having adenine and guanine as our examples. And one more we are having is the pyrimidines. We are having the uracil, thymine, and also cytosine, right? Yes. So all of these three belong to the pyrimidines, and only the guanine and adenine belong to the purines, right? So now we are having nucleosides and also the nucleotide. Okay. The ultimate difference between the nucleoside and nucleotide is if in the case of nucleoside, only a nitrogenous base and a sugar makes the nucleoside. But if you add phosphate molecule, it will result in the nucleotides, right? So generally our DNA molecules and one more we are having is the RNA molecules are made up of the nucleotides only. Okay, so we are having some examples, that is the adenosine, guanosine. Okay, these are all examples for the nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, and also the uridylic acid, etc. are example for the nucleotides. Okay, I have not mentioned it in the presentation, but you can make it out what I'm trying to tell over here, right? So we have discussed the first part of the chapter, and what we can now do is we can look into some of the important questions for the this chapter. The first question what we are having is which of the following is not true? Trichloroacetic acid is used as an agent to grind the tissues. The earth crust and the tissues have same constituents. The tissue contents are only acid soluble. The acid soluble pool has organic compounds. Okay, so you can make it out over here that the trichloroacetic acid is used as an agent. Okay, we can say a buffer or an organic solvent, we can call it in a more appropriate way. For the grinding of tissues. Exactly, this statement is actually true. We use trichloroacetic acid, or in some cases you also use any other complex organic compound for grinding the tissues. Okay, and earth crust and the tissues have made up of same constituents. Exactly, right? Carbon, oxygen, hydrogen, nitrogen. You do find these constituents in both the living cells and also in the case of earth crust. And finally we are having the third statement, that is the tissue contents are only acid soluble pool. No, that is not the case, right? So hence C is our right answer, because you find two fractions, that is the acid soluble pool and also acid insoluble pool. The acid soluble pool is mainly, it's present in the filtrate, and the remaining solid precipitate is present in the muslin cloth, is actually acid insoluble pool, right? You don't find only acid soluble pool, you find two pools, that is the acid soluble and also acid insoluble. The last statement, obviously it needs to be true, that is acid soluble pool has organic compounds. Exactly, it does have the organic compounds. So the right answer, that is the not true statement, is the C option over here. Now the next question, which of the following is least in the earth's crust, right? So now you people have to go to the table and try to refer which of the following is very least in the earth crust constituent, right? Nitrogen, carbon, silicon, and hydrogen, right? The very little constituent what they have mentioned in your textbook is the nitrogen. So hence the right answer is going to be what? Nitrogen, right? Yes, silicon is the second highest constituent, we all know that. So hence the right answer for this question is A, nitrogen. Yeah, so let me remove this. Yeah, but let me mark the answers. C, and here we are having A. Exactly. Now let us look into the next question, that is the third one. Which of the following technique is used for extraction and purification of compounds? Electrophoresis, chromatography, page, that is a polyacrylamide gel electrophoresis. Let me write this: polyacrylamide gel electrophoresis. Right, now we use this technique mainly for separation of proteins, remember. Okay, we mainly use this for separation of proteins. And the last one we are having is the centrifugation, right? So we have mainly mentioned the term purification, though all of the techniques, they are involved in separation and extraction, but the purification is mainly done by the chromatographic techniques only, right? Hence the right answer we are having is B, that is chromatography. Now, the next question. The probable structure and molecular formula can be determined by the physical techniques, chemical techniques, biological techniques, analytical techniques. Okay, so if you look into the previous question, we have mentioned about the chromatography is mainly used for extraction and purification, right? So now chromatography is an example for analytical techniques. Okay, analytical, the name itself indicates for analysis. Okay, and the analysis is mainly used for determining a empirical structure and empirical formula, molecular formula of that particular substance that we are interested in, right? Hence the right answer over here is the D, that is analytical techniques. Probable, what do you mean by probable? Probability, okay? Maybe or may not be, okay? That appears to be true initially, or it may be even false later on, right? Yes. So hence the D is our right answer. The next question, drying and destructive methods such as incineration, that is burning, is used to determine the dash of tissue, that is organic constituents, inorganic constituents, biochemical constituents, all of the above. It is clearly indicative that inorganic constituents, that is sodium, calcium, magnesium, and so on, they are mainly determined by drying the tissue and by burning the tissue. And finally, the ash that remains is mainly used for what we can say, the determining the inorganic constituents. So now let's move on to the next question. The element that is highest constituent in the human tissues and earth crust is silicon, nitrogen, oxygen, carbon. So we have to again look into the chart, and as I had emphasized quite importantly, that oxygen is the highest of all, right? So hence C, oxygen, is going to be our answer for this. That is the highest constituent in both the human tissues and also the earth crust is the oxygen. Now, let's look on to the next question. Which of the following statement is true? Silicon is the least of all constituents in the human tissues, and carbon is the highest constituent in the both earth's crust and also the human tissue. And grinding of tissue with trichloroacetic acid is used to determine the inorganic constituents of the tissue, and inorganic elements belong to acid insoluble. So now let us try to recall all the points that we have made till now. So it's clearly indicative that inorganic elements belong to acid soluble, not for acid insoluble, right? So hence we can say that the D statement is actually false. The next one we are having is grinding of tissue with the trichloroacetic acid is used to determine the inorganic constituents. No, right? We mainly use this method for determining the organic constituents. Hence, even the third statement, that is C statement, is also false. The second statement, you can see that the carbon is the highest constituent in both the Earth's crust and human tissues, right? So you can see that it is actually the oxygen that is the highest constituent. Carbon is the second highest constituent only in the case of human tissues, but not in the case of Earth's crust. So this statement is again false. The first option we are having is silicon is the least of all constituents in the human tissue. Exactly, it is the very negligible that they have mentioned in your textbook. So hence A is going to be our right answer for this question. It is the truth statement. The next question, which of the following statement is not correct? The elemental analysis is used to determine the inorganic constituent, and the ash contains the inorganic elements. The functional group such as ketones, aldehydes, etc. are the biological point of view, and carbon derived compounds are called as biomolecules, right? So now again they're asking this time about the false statement or incorrect statement. The carbon derived compounds are called as the biomolecules. Exactly, it is actually true. And the functional groups such as the ketones, aldehydes, etc. are in biological point of view. No, The classification based on the functional groups is actually the what we can say biological point of view, but determining the functional groups is actually the chemical or chemistry point of view. So hence we can say the C option is not true. We can directly confirm that C is our answer, right? So hence C is our answer. And the B statement, that is ash, mainly contains the inorganic elements. Exactly, it is true statement. And elemental analysis is mainly used to determine the inorganic constituents. Exactly, even that is also a true statement, right? So hence the right answer for this and the not correct statement we are having is C, or the third statement. The next question we are having, the net charge of this Zwitter ion is 1230. It is simple. We have mentioned in the earlier class that the charge is going to be 0. The net charge is going to be 0. It carries no net charge, right? So hence D, that is 0, is our answer. The next question, the acidic group in the amino acid is the nitro group, carboxyl group, amino group, hydroxyl group. We all know the carboxyl group, that is a COOH. Okay, the COOH, that is the carboxyl group, is mainly giving the acidic group, and the amino group is mainly giving the that is NH3 is the amino group, or NH2 group. It mainly gives the basic property to the amino acids. So hence B, that is carboxyl group, is our right answer. Now moving on, which of the following is an aromatic amino acid? Right? Which of the following is an aromatic amino acid, right? Tyrosine, histidine, arginine, lysine, right? So now we know there are 3 examples that have been mentioned in a textbook: tyrosine, tryptophan, and phenylalanine, right? Only these 3 belong to the aromatic amino acids, right? And hence A is our right answer. Histidine is actually positively charged, okay, and arginine is also positively charged, lysine is also positively charged, right? So all these are actually positively charged amino acid. Now, moving on, match the following. We are having 2 columns, that is column 1 and column 2. The first column we are having is the first one, methyl group, hydroxymethyl group, acidic amino acid, and a neutral amino acid. The column 2 we are having is the glutamic acid. The next one we are having is the alanine, and the valine is the third option. Last we are having is the serine, right? So we all know the methyl group is mainly present in the case of the alanine, so we can write 1 number over here. Hydroxymethyl is mainly present in the case of serine. The fourth option will be having the second one. And the acidic amino acid we are having is the glutamic acid is an example for acidic amino acid. The neutral, the last remaining we are having is the valine, hence the fourth will go over here. So if you look into the options that has been mentioned over here, the first one matches with the second option exactly, and yeah, so we can see that the second option is similar to what we have mentioned over here. B is going to be the right answer. 1 with 2, 2 with 4, 3 with 1, and 4 with 3, right? Yes, so this is going to be our right answer. Moving on, the next question, which of the following statement about the amino acid is true? The alpha carbon has a 3 different group attached to it, and if the R group is hydrogen, then the amino acid is alanine. Okay, so I think we have missed a word over here, that is amino acid. Okay, the amino acid doesn't change, the charge of the amino acid doesn't change irrespective of the pH in which it is present. I think this is what the option should be, and there are 20 amino acids occurring, right? So they say that there are 3 different groups are attached to it, so we can say that it is actually 4 different groups are attached to the alpha carbon. And if R group is actually hydrogen, the amino acid is called as alanine. No, it is called as glycine, right? So hence a B statement is also not true. And the third statement, the charge of the amino acid doesn't change irrespective of the pH in which it is present. No, it actually changes according to in which pH condition it is present. Even C option is also false. And in the D option we are having is there are 20 amino acids occurring. Exactly, we are having 20 amino acids. So if you refer any higher biology textbooks such as mainly biochemistry and all, they even mention that there are 21 amino acids. Okay, and the 21st amino acid is called as this selenocysteine. which is a modification or derivative of serine amino acid, if I remember properly, right? So I think the right answer over here is the D option, that is there are 20 different amino acids occurring. That is going to be our true statement and the right answer for this question. Now moving on, which of the following is a lipid? Fatty acid, diglycerides, phospholipids, all of the above. Yes, exactly. All of the three mentions are actually the lipids. Diglycerides are also derivative of a lipid. Fatty acid is a simple lipid. Phospholipids are also derivative of lipids, glycerols, etc. Right? So we can say all of the above are the lipids. Now moving on, the total number of carbons in the arachidonic acid is 18, 20, 25, 15. We know in the case of palmitic acid it is 16, and in the case of arachidonic acid it is 20. Hence B is going to be our right answer. The next question, a nucleoside is sugar plus phosphate, phosphate plus nitrogenous base, nitrogenous base plus sugar, and nitrogenous base only. So a nucleoside is mainly made up of a sugar and a nitrogenous base, right? If you attach a phosphate molecule, it becomes a nucleotide, right? So hence the C is, that is a nitrogenous base, and sugar plus a sugar will make the nucleoside. So C is going to be our right answer. The next question, which of the following is not true about lipids? Palmitic acid has 20 carbons. Exactly, it is not having 20 carbons, it's actually having 16 carbons, right? So you can see that A, the first option itself, is not true. So hence A is going to be our right answer. But let us check on to the other three options also. The arachidonic acid is a fatty acid, and oils have a lower melting point, and fatty acids are esterified with glycerol to form monoglycerides. Exactly, D statement is also true, C is also true, and arachidonic acid is a what we can say fatty acid. It is a polyunsaturated fatty acids. So the B, C, D are actually true statements, but the A statement, that is palmitic acid has 20 carbons, it should be having 16 carbons, right? So hence A is our not true statement and the right answer for this question. Now moving on, an example for phospholipid is the fatty acid, gingerly oils. Next one we're having is the palmitic acid. The last one we are having is the lecithin, right? So lecithin is an example for the phospholipids, and lecithin is actually found in the membranes, cell membranes. Remember this point. So D is our right answer. The next question, the lipids with complex structures are found in gastrointestinal tract, neural tissues, sensory system, all of the above. Okay, lipids with complex structures. Example we are having is the ceramide or sphingolipids, etc. Right? So all of them are actually complex lipids, and you have mentioned, like you have read it, that they're mainly found in the neural tissues. Hence B is going to be our right answer. Next question we are having is the DNA and RNA consists only of the nucleotides, nitrogenous bases, the sugars and phosphates, and the nucleotides, right? The first option is nucleosides. Sorry, I mispronounced it. So we all know the DNA and RNA are ultimately made up of the nucleotides only. Hence D is our right answer. Nucleotides is going to be the right answer. So we have discussed around 20 questions from the first part, and I guess it is sufficient for this particular session. What you people can do, as I suggest always, okay, and I will be suggesting it every time in every video, that you people have to go and try to solve more and more questions. In that way, you can try to increase your accuracy in the questions. Don't feel that the questions are very easy, that what we have discussed. The questions are always framed in this particular way itself. And as I have said that at the end of the complete of whole unit, that is all these the 3 chapters of the cell, we will be discussing the previously appeared questions, right? So let's meet in another video with the next part of biomolecules. Thank you.